Hello everyone, I'm Apoorva Shankar, Assistant Professor from the Department of Life Sciences, Kristu Jayanti College. Today let us discuss about the ultrastructure of the fungal cell. The fungi can be a single-celled or a very complex multicellular organisms. The fungi are ubiquitous in nature. They are distributed in soil, water, and in land. A group majority of the fungi are decomposers in nature, and they behave as saprophytes, feeding on the dead and decaying matter. While a few of them are parasitic in nature, they can cause diseases in plants and in animals. In humans, one of the common fungal diseases which we all are aware is the athlete's foot and ringworm and thrush, which are caused by the fungi. The ultrastructure of the fungal cell, the different components we'll be discussing here. Firstly, the cell wall. The cell wall of the fungus is made up of, there is slime molds which lack a true cell wall or a rigid cell wall or the cell organelles, and they belong to the kingdom Myxomycetes. And there is another group of fungus which are called the true fungus, where the cell wall is made up of chitin, cellulose, or glucans. Are present in the cell wall in the form of layers, and the cell wall is very important in terms of reproduction, recognition, and deception. The fungal cell wall. This is the structure of the fungal cell wall. It contains a cell membrane with various membrane proteins, a protective layer of the chitin, as well as glucans, manoproteins on its surface, and different cell walls contain different types of glucans. The plasma membrane. In fungi too, the cell wall is followed by the plasma membrane that encloses the cytoplasm. The plasma membrane is semi-permeable in nature, and it is very similar to that of the prokaryotes. There are specialized organelles in the surface of plasma membrane where the fusion of secretory vesicles of cytoplasm occurs. The plasma lemma invaginates and forms a pouch-like structures that enclosing the granular or vesicular materials within them. After the plasma membrane, we have the cytoplasm. The cytoplasm and most organelles are inclusions of the fungal cell wall are present in the cytoplasm, which is a typical of a eukaryotic organism. The cytoplasm is a colorless and it is sap-filled with vacuoles are found in cytoplasm. Cytoplasmic inclusions are dead, they're non-functional and unimportant for the fungal survival. Cell organelles are endoplasmic reticulum, mitochondria, ribosomes, Golgi bodies, and vacuoles are present. Lamosomes are also present in the fungal cell walls between the plasma membrane and the cell wall. So this is the structure of the fungal hyphae, showing the ribosomes, lamosomes, which are present beneath the plasma membrane, cell wall, nucleus, endoplasmic reticulum, vacuoles, glycogen, and mitochondria are visible in the structure of the hyphae. The mitochondria of the fungi are clearly recognizable. They have a double bilayer membrane and they contain complex internal membranes. They differ from other eukaryotic organisms in that mitochondria commonly elongate, oriented along the hyphal axis. The mitochondria is the powerhouse of the cell. It is the machinery for the transcription and translation of the organelle-specific DNA. The fungal cell wall structure, showing the cell wall, cell membrane, Golgi apparatus, nucleus, mitochondria, rough endoplasmic membrane. The endoplasmic reticulum. The presence of endoplasmic reticulum in a fungal cytoplasm is observed through an electron microscope. It is made up of a system of microtubules with the small granules. In most of the fungi, it is highly vascular. It is loose and irregular as compared with the cells of green plants. In multinucleate hyphae, the nuclei may be connected by the endoplasmic reticulum. The vacuoles, which you see, the bigger vacuoles in the hyphal fungi, they are essential for cell function and are characterized by the presence of spherical to tubular vacuoles. They are found in the older cells of the hyphae, and the end of the hyphal tip of the young hyphae lacks vacuole. They are surrounded by a membrane called tonoplast. They help an important role in osmoregulation. The Golgi apparatus of the dictyosomes. Except in oomycetes and non-fungal eukaryotic cells, Golgi apparatus is rare occurrence in the fungal cells. In oomycetes, Golgi apparatus consists of stacks of folded membrane functioning in secretion. Major function is to process and package macromolecules and transportation of lipids around the cell. Other inclusions, the cytoplasm contains various kinds of inclusions, examples of stored food or lipid globules, granules of glycogen, oils, and other carbohydrate trihalose, proteinaceous material, and volutin. There are no starch grains of pigments, they lack chlorophyll. This is very important. A fungus lacks chlorophyll, unlike algae, they cannot prepare their own food. Nucleus, the cytoplasm in the individual cells contain one, two, or more globus ellipsoid nuclei, which in the somatic proportion are small and usually range from 1 to 3 micron in diameter. They consist of a central dense body with a clear area around it, chromatin strands, nuclear membrane. It has a nucleolus. The chromatin strands become organized into chromosomes during nuclear division. The fungal flagella, the higher fungi lack motile cells in the life cycle, with the exception of zygomycetes. There are characteristics of the lower fungi. The motile cells in the lower fungi are furnished with one or two fine protoplasmic whip-like threads known as flagella. These flagella are thin hair-like emergence of the cell cytoplasm, which functions in propelling or locomotive structures of the cell. There's a single anterior or posterior flagellum, or they can occur in pair in the cell. So this is an example of a flagellum with whiplash flagellum and tensile flagellum, thin hair-like delicate structures which are found in lower aquatic fungi. Apart from this, the fungi can be septate or aseptate in nature. The protoplasm may form a continuous uninterrupted mass running the length of the branching hyphae, or the protoplasm may be interrupted at regular intervals, forming cross walls called the septa. The septa divide up the hyphae into individual discrete cells, which are interconnected by the hyphal compartments. There may be various types of septas which are present in different fungi. They are complete septa, perforated septa, dolipo septa, etc. The function of the septa is that it provides a structural support and also the first line of defense when the part of the hypha is in damage. It also facilitates differentiation in fungi. Here are the different types of septa examples. Oomycota and Zygomycota, they have a complete cross wall septa. In Basidiomycota, we can see the presence of doliposeptum, which has a parenthesomes in the end, that is the swelled up endings. And Ascomycota and some mytosporic fungi, we can see the presence of voronin bodies. These voronin bodies, whenever they are present around the septal pore, and whenever there is a damage to the hypha, the voronin bodies go and close the septal pore in order to protect the hyphae of the fungus from the leakage of the cellular contents. Some other mytosporic fungi can have microspores with multiple pores within a single hyphal wall. Different types of spores. There are different types of asexual spores. Fungi produce two major types of asexual spores. They are called the sporangiospores and the conidia. The sporangiospores are endogenous, formed and contained within the sporangium, formed as a result of the cleavage of protoplasm around the nuclei, followed by a result of the cleavage of the protoplasm around the nuclei. In some cases, the formation of the wall around each nucleate proportion. There are two main types, zoospores, which are motile, and aplanospores, which are non-motile. Aplanospores are non-motile extracellular spores formed within the cell, the wall of which is distinct from the other parent cells. Zoospores, these are the motile spores, motile sporangiospores, and the sporangia in which they can form a zoosporangia. The zoosporangia in chytidomycetes, they have a single posterior smooth whiplash flagellum. In case of hypocytidomycetes, you can see a tensile flagellum. And in case of Eumycota, you can see a biflagellate with a tensile and whiplash type of flagella present. Because the zoospores are motile, the fungi that produce them will require water at some stage during their life cycle. Three different types of zoospores are distinguishable, that is the Chytidomycota, Hypocytidomycota, and an Oomycota. Conidia, the conidias are usually exogenous in nature, often formed on the tip of the supporting hyphae called the conidiophore. Conidia can develop in variety of ways. The characteristic of metosporic fungi and fungi belonging to Ascomycota and Basidiomycota form conidia. There are two main types, thallic and blastic. The thallic conidia develop by the septation and fragmentation of a hyphae and may develop at the tip of the hyphae or in an intercellulary position. In both the cases, all the layers of the hyphal wall are involved in the spore formation. It is further divided into two types, orthospores and chlamydospores. Orthospores, they are formed by the septation and fragmentation of the existing hyphae. The elements of the hyphae, inclusive of all the layers, become converted into the conidia. Each fragment is rounded off and liberated in succession. The separation of the conidia from one another is due to the breakdown of the middle region of the each septum. So here the entire hyphae in arthrospores gets uh, gets separated or fragmented into small spores called orthospores, which individually can germinate into a hyphae. Next one is the chlamydospores. This is a type of a resting or a survival spores which are formed during the unfavorable conditions. An intercalary and apical hyphal cell, the compartment enlarges around and develops a thick, often pigmented wall. It contains dense cytoplasm and nutrients, which are storage compounds. All the wall layers are involved in the formation of the chlamydospore. They become isolated from the adjacent hyphal compartment by the sealing of the septal pores. Usually, they develop under stress conditions or unfavorable conditions, and they are helpful for the survival during the unfavorable conditions. And when favorable conditions return, the chlamydospores then germinate into a new hyphae. Plastic conidia, they develop by budding or swelling process and may develop as a single spores in the succession to form a chain of spores. The blastospores formed by the budding of a hyphae or the yeast cell, both the wall layers are involved here and the spore may remain attached and bud further blastospores giving rise to the branch chain of spores. Porospores, they develop spore emerges through a distant pore in the hyphal wall. Only the inner layer of the hyphal walls is involved in the spore development. The new spore then develops into an own inner layer, wall layer. The outer spore wall is often thickened and pigmented. A scar is usually obvious at the point of detachment from the hyphae, that is the conidiophore. Aluriospores develop as a single terminal spores. Conidiophore apex inflates and becomes separated by a septum at an early stage in the spore development. Both wall layers are involved in the spore formation. The spore possesses a wide, truncate scar. Normally, no further development of the spores occurs at the point of detachment. So the next spore usually has to develop by the production of branch below the scar on the conidiophore. Ankylospores. In some species that form conidia in a manner similar to that described for aliospores, a new growing point develops at the scar and a chain of spores may develop. The conidiophore gets a little longer with each spore produced. Annellations, that is the ring-like scars, are observed around these elongating proportions. Each annellations represent the production of one ankylospore. Hyalospores form in succession. Each spore is pushed up from the tip of the conidiophore, which is now called a filidae. The spore wall is nude and distant from both wall layers of the filidae. The first spore has a cap which represents the tip of the filidae wall through which the spore emerged all other spores in the chain and are smoothly rounded in nature. Since fungi are often classified according to their spore-producing structures, these spores are often characteristic of a particular taxon of a fungi, like the zygospores. These zygospores are produced by the zygosporangium, which is characteristic of zygomycetes, and ascospores are produced by an ascus, characteristic of ascomycetes. Basidiospores are spores which are produced by the basidium, which is characteristic to basidiomycetes. Asciospores are produced by the acium in some fungi such as rust and smuts. Uridinospores are produced by uridinum in some fungi such as rust and smuts. Teleospores are produced by telium in some fungi such as rust or smuts. Oospores are produced by oogonium, which is characteristic of oomycetes. Sexual spores, these are sexually produced spores of higher fungi, results from meiosis and are formed either in sac-like structures, asci, which is typical of ascomycota, or on the surface of the club-shaped fungal structures, which are called basidia, which is typical of basidiomycota. Asci and basidia may be born naked or directly on the hyphae or in various types of sporophores called ascospores or the basidiocarps, depending upon whether they bear the asci or basidia respectively. That was about the ultrastructure of the fungal cell. Thank you.